Great. So I'm back. So now we're going to look at the antibody and the structure of the antibody. And the structure of the antibody is really important for its function and also the classification of the antibodies into different types. So antibodies, they are formed by two heavy chains. So these are called heavy chains because they are big. So we have these two heavy chains. And we are also going to have two lighter chains, two light chains. They are called light chains because they are smaller than the heavy chains. And the heavy chains, they are going to be interacting with each other through these disulfide bonds. Similarly, the light chains, they are going to interact with the heavy chains also through these disulfide bonds. So the antibodies have two different segments. So an antibody has here this constant segment that is going to be the same regardless of the infection. And an antibody is also going to have here a variable segment. So this variable segment is going to be very specific for different antigens depending on the type of infection that we have in our bodies. And in the variable segment, we have a very important part, which is called the antigen binding site. So this is going to be the specific part of the antibody that is going to interact with a specific antigen. And this antigen binding site is very specific and it changes depending on the different type of virus or the different type of bacteria that we have to defend ourselves from. So depending on the constant segment, we are going to look at different 5 types of antibodies that we have in our bodies. So the first type that we are going to look at are the IgGs. So IgGs are around 80% of the antibodies in our system, and they are going to defend us from bacteria, bacterial toxins, and viruses. So these are the antibodies that are going to participate in the processes that we're going to see in a minute. And also they can cross the placenta for giving this fetal immunity to transfer the immunity from the mother into the fetus during pregnancy. Also, we are going to look at these IgEs. So these IgEs, they are going to be antibodies. Their structure is very similar to the IgGs. So the IgG structure is what we have just seen. So we have here the heavy chains and the light chains. And the IgEs, the structure is also very similar to these IgGs. So these IgEs, they are going to interact with basophils and also with mast cells for the secretion of histamine. So if you remember from the previous lecture, histamine is really important for the early inflammation, and it's also very important for allergies. Then we have another type of antibody, which is the IgD. So the IgD structure is also similar to the IgG. And the IgD is important because IgD is the antibody that is on the surface of the B cell for the process of B cell synthesization. So this is how the B cells, they are going to bind to the antigen so they can start the process of activation and then the process of division and differentiation for differentiation into plasma cells and the production of the antibodies. Then we also have the IgMs. So the IgM structure is very different and is this pentagon in which we have 5 different antibodies okay. And these IgM are going to be the first antibody that is secreted when we encounter an antigen. And then finally we have the IgA. So the structure of the IgA is kind of 2 IgGs together like this. And this IgA is going to be secreted through the mucus and also through tears and saliva, for example. And they are going to encounter the antigens and the different microorganisms before they enter in our body because they are secreted outside. So they are going to be kind of a barrier before the antigen can enter into our body, okay? So the secretion of these IgGs depends on the different type of adaptive immune response, and we have 2 different adaptive immune responses in which antibodies are important. So first of all, we have the primary response. So the primary response happens the first time that we encounter an antigen. So in here, we are going to have the time in weeks. And here we have the antibody concentration in the serum. So during the primary response, we're first going to have the IgM here. And the production of the IgM is going to peak around 1 week after the infection. And then after the IgM, we are going to have the production of IgG, which is going to peak around 2 weeks after the exposure to the antigen. This is why, for example, when we get vaccinated, the maximum exposure or the maximum efficiency to the exposure of an antigen happens after 2 weeks of the injection. And then also we have the secondary response. So the secondary response happens when we encounter the antigen for the second time. So we already have the memory B cells to fight this antigen. So if here we have the concentration of antibodies in the serum, and here we have the time in weeks. First, we are going to have the IgM, which is going to peak the same around 1 week. But then, because we already have the memory cells, the production of the IgG is going to be very quick and almost immediate at the same time as the IgM. In here, in the primary, because it's the first time that we're getting exposed to an antigen, we don't have those specific antibodies yet, and we need to give the B cells some time for the process of sensitization, activation, and then differentiation. That's why it can take up to 2 weeks to generate this IgG. But here in the secondary response, we already have the memory B cells, so they start producing IgGs really quickly for that specific infection. Okay, so finally, we are going to look at the process of the different functions of the antibody. So we are going to look at 5 basic functions of the antibody. So give me a sec that I delete a little bit here. Okay, so finally, we are going to have a look at the different functions of the antibodies. And we are going to look at 5 main functions of antibodies that are going to allow us to fight infections, bacterial toxins, bacteria, and viruses better. So the first function of the antibodies is the neutralization. So let's say that we have a bacterial toxin or a virus, and this bacterial toxin or virus is going to express some antigens, right? So then the antibodies, they are going to bind to these antigens, so the bacterial toxins or the virus cannot bind to other tissues or cells in our body. So it's going to neutralize here the antigens, and this is going to prevent the binding or the interaction of the viruses or the bacterial toxins with other cells in our body. The second function that we are going to look at is the function of precipitation. Precipitation and agglutination. So the antibodies can bind to many different, to more than one antigen, or to more than one particle at the same time. So the same antibody can bind to more than one particle at the same time. And then these toxins or viruses that were soluble in our plasma, in our blood, or in our interstitial spaces, thanks to this, they are not going to be soluble anymore. So they are going to precipitate because the antibodies, by binding to more than one antigen or to more than one particle, they are going to form these big immune complexes that are not going to be soluble, and this is why they precipitate. And also this process by which we create these immune complexes is called agglutination, okay? Agglutination. Then also, their function is to initiate the process of phagocytosis. So the antibodies, they are going to attract different cell types, different immune cell types that are going to achieve the process of phagocytosis. So they are going to englobe and digest these different toxins and viruses. So it's going to attract 2 different cell types that are very important. It's going to attract the neutrophils. And also is going to attract the macrophages. That are going to be the cell types that are going to phagocytose all these different toxins, bacteria, compartments, and also viruses that are identified by the antibodies. Then very related to the process of phagocytosis, also we have the function of opsonization. So the presence of the antibodies is going to facilitate the process of phagocytosis, and this is what opsonization means. So this is going to make this process by the neutrophils and the bacteria much easier, because they are going to be able to grab to these toxins much better. And finally, the last function is the activation of the complement system. Okay, so these are the 5 different functions for the antibodies. So we have gone through everything with antibodies in this lecture today. We have gone through the production of the antibodies by the B cells, also the structure of the antibodies, the different types of antibodies, and also the primary and the secondary response, and finally the different types of antibody functions. So I hope that you enjoyed this lecture. I'll see you next week. Thank you.